Hello, thank you all for joining me. My name is Javi Sala, and I'm a PhD student at the National Museum of Natural Sciences in Madrid, in Spain, with Borja Milá. And today I'm bringing you a study titled Evolutionary Mechanisms Across the Postglacial Radiation of the Janko. So in our group, we are interested in understanding the origin of evolutionary divergence, and particularly in the relative role of drift against selection, on the role of local adaptation in ecological speciation models, and on how historical demography influences these mechanisms on the early stages of divergence. To assess these processes, we must study speciation as it happens or shortly after, so we can still detect the footprint of selection before other differences accumulate across the genome. Recent radiations typically span the entire continuum, with different taxa showing varying degrees of reproductive isolation, and thus provide a unique opportunity to study the adaptive process of speciation from the very loci under divergent selection to the formation of independent lineages, linking patterns of genomic and phenotypic variation from the population to reproductively isolated lineages. A great example of recent radiation is provided by the songbird genus Janko, a fascinating case of speciation in action, from local populations to species, and from fully isolated to freely interbreeding parapatric forms. The genus is currently grouped into 5 species. However, the most intriguing aspect is found in the dark-eyed Janko, which inhabits conifer and broadleaf forests across temperate and boreal North America. So more or less, everything above the Mexico and the US border is dark-eyed Janko, more or less. So despite substantial geographic and phenotypic variation, it is considered a single species due to the lack of reproductive barriers. Also, their forms interbreed in the contact zones. Weak reproductive barriers are mostly due to the extremely recent nature of this radiation. So after the last glacial maximum, 20,000 years ago, a lot of populations expanded their ranges, a lot of animal populations, including birds. But the Janko case stands out as unique. As the yellow-eyed Janko, Janko phaenotus, here displayed in yellow, extended its range to the north, it gave rise to the dark-eyed Janko, which subsequently diversified into at least 5 distinct forms with contrasting plumage coloration. And all of this just within the last 12,000 years, as can be seen in this mitochondrial DNA phylogeny, where you can see that the upper clade has large and shallow polytomy that encompasses all the dark-eyed Jankos and the yellow-eyed Jankos. So up to this date, we could not solve the phylogenetic relations among these taxa. Our group has done extensive field samplings in North and Central America, capturing Jankos one by one to extract tissue samples, and we also visited museum collections to gather data on morphometrics and color. So the dark-eyed Janko are currently grouped into 3 main lineages according to male plumage coloration. We have the Yemalis group, the Oreganus group, and the Canitheps group. As a fun fact, if you go birdwatching around Montreal these days, you might probably see Yemalis individuals. So we know that sexual signaling traits diverge at a faster rate than ecologically related traits. See how in this discriminant function analysis, the dark-eyed Jankos display more structure in the color DFA than in the morphology DFA, suggesting a prominent role for sexual selection in promoting phenotypic differences and divergence. This is why this radiation is often considered an example of non-adaptive radiation. But we want to know if other mechanisms are also involved in it. So our goals here are to reconstruct the evolutionary history of the genus, to understand the relative roles of natural selection, sexual selection, and drift in the dark-eyed Janko radiation. Also to identify the genomic architecture of differentiation between the 3 male lineages, and to explore the genomic basis of fitness traits. So we've performed genome resequencing of 240 individuals at 18x coverage, and we used a high-quality reference genome to map the sequenced reads. And well, as you can see, our sampling is extensive at multiple levels. So the whole genome data has provided us much greater resolution to detect structure within the dark-eyed juncos and the evolutionary relations among taxa. We used Astral to estimate a species population tree based on 32,000 gene trees. And the resulting tree reveals a consistent northward succession of the taxa, which is a huge step for us. So the yellow-white juncos from Mexico gave rise to the dark-eyed juncos in Arizona, then the Rockies, the West Coast, and finally the boreal taxa and the Appalachians. And well, so for the first time, the phylogeny provides us with a well-defined topology of the diversification during the expansion, and also supporting or aligning with the post-glacial expansion hypothesis. If we shift our focus to the analysis of the population structure, we can see that in an admixture analysis, K2 sorts taxa latitudinally, so again, aligning with the post-glacial expansion hypothesis. But when we look at K6, the suboptimal value of K, we see that the populations of Pinosus, Townsendi, Dorsalis, and Carolinensis display a strong evidence or strong differentiation compared to the rest of the taxa. These populations have in common that, to some extent, they display a degree of isolation on their breeding ranges. But we want to know what are the mechanisms underlying this strong differentiation. So we used genome-environment association analysis, specifically redundancy analysis, which allows us to model linear relations between environment predictors and genomic data. And we see that the aforementioned populations now exhibit association with the environment predictors. However, when we perform a partial RDA, where we use the neutral structure as a covariable in the model, and thus controlling for the effects of neutral structure, the small and isolated population of Townsendi collapses in the middle of the plot. This suggests that its strong differentiation is due to non-adaptive processes, that is, genetic drift. On the other hand, the highly differentiated populations still exhibit strong association with the environment predictors, suggesting a role for local adaptation on their differentiation. We are also interested into understanding the genetic architecture of differentiation between the three main distinctly colored dark-eyed lineages. And here I'm displaying an FST plot between those lineages, and we've marked in red the private outliers and in blue the shared outliers. So first thing to notice is that most outliers are located in shared regions, while private outliers are scarce. And also, well, to further understand these patterns, we assessed the relation between FST and recombination rates for all three comparisons. And in all cases, we found a negative relation, somehow expected, but the interesting thing is that we found very few outliers in high recombination regions. So this suggests that low recombination regions are crucial hotspots of differentiation, which are shared across lineages, and it is where selection is acting. And also that selection might not be strong enough to fix beneficial mutations on high recombination regions. So we've also looked at the genetic basis of fitness traits. One of the fitness traits we're interested in, and the trait I'm bringing to you today, is color variation. We have used a multivariate linear mixed model with GEMMA, using the color as the response variable. And you can see the results of this GOS in the upper Manhattan plot. So we recover 5 highly associated regions in the sexual chromosome Z. But when we perform a selective sweep analysis on this chromosome on all lineage comparisons, we see that the region highlighted in red is the only one that overlaps with a region of recent positive selection. Moreover, when we looked at the functional annotation of this region, we found that 4 out of the 8 genes had already been reported to be involved in pigmentation in a chicken breed. So we think this region is key to understand plumage color variation and its role during the diversification process, which is the main difference between the 3 lineages. Now we're looking into the role of structural variation, And using local PCA, we found a large chromosomal inversion in chromosome 5. If you look at this plot, you can see a 20 megabase region highly associated with the principal component 1 of multidimensional scaling. And when we perform a PCA of the whole region, we see that the PC1 sorts taxa in 3 groups. And when we plot that against the heterozygosity, we see the classic pattern of an inversion with a homozygotic wild type, heterozygotic group, and a homozygotic group for the inversion. But what's even more interesting is that we found a positive correlation between the prevalence of the inversion and the latitude, with the inversion being a common feature, a widespread feature among the dark-eyeds, and being pretty scarce, almost inexistent, in the yellow-eyed Jankos. And when we perform a geo-enrichment analysis of the whole region, where we got 437 genes, the most significant geoterms turned out to be muscle function and regeneration, circadian rhythm regulation, and behavioral responses. So these functions suggest that this region could be somehow related to the migratory behavior, which is also a characteristic, a feature of the dark-eyed Jankos that migrate. Yellow-eyed Jankos do not. We also have here a geoterm related to DNA UV damage repair, and that it can be also correlated with latitude. You know, more latitude, more exposure to UV lights. So we think this inversion, this structural variant, has an adaptive component or in this diversification. So I'd like to highlight that the phylogenomic data has supported the postglacial expansion hypothesis. Both adaptive and non-adaptive processes turned out to be crucial in the diversification of the dark-eyed Janko populations. Regions under selection were found in low recombination regions mostly. A region in sex chromosome Z potentially underlies plumage color variation between the 3 distinctly colored lineages. And it seems that structural variation has had an important role in local adaptation during the postglacial diversification. And I want to thank my colleagues at the Mila lab, the von Holt lab in Princeton, and Maria and Guillermo, the former lab members. Sure. Mm-hmm. Yeah, actually, this is the pattern that we found. Yeah, for example, we kind of know that in chromosome 1, there's a centromere in this highly differentiated population region. So yeah, you're kind of right with it.
Hi, everyone. I apologize in advance for looking like a talking head. Thank you for coming. My name is Megan, and I'm here showing you a portion of some ongoing work involving a number of collaborators looking at the phenotypic divergence and local adaptation of eastern gray squirrels in urban and rural environments. So the striking transformations that happen to the natural environment that come with the expansion of cities offer this unique opportunity for us to deepen our understanding of rapid evolution to novel habitats. So within cities, changes in behavior, morphology, and thermal stress have been well documented in taxa within the cities. However, identifying the evolutionary causes to these changes remains a bit more difficult, particularly when we're trying to identify the genetic drivers associated with this phenotypic divergence along urbanization gradients. The mechanisms underlying urban and rural trait variation can be non-adaptive, resulting from strong genetic drift due to founder effects or low effective population sizes in cities. They can also be local adaptation can also cause genetic variation along urban-rural clines. But the different modes of selection that contribute to urban evolution are often underappreciated. So urban adaptation is often the leading hypothesis explaining phenotypic divergence between urban and rural areas. And previous studies have identified a number of genomic regions associated with local adaptation to novel selection pressures within the cities. Conversely, the urban environmental changes could potentially weaken selective pressures that still remain relevant within the within the rural environment, resulting in sort of a relaxed selection occurring within cities. So our project is to understand whether what sort of modes of selection are acting on the genomic and phenotypic divergence of eastern gray squirrels in urban environments. So eastern gray squirrels are native to eastern North America, but have been introduced to western North America, Europe, South Africa, Australia, and while they were historically restricted to rural woodlands, have now been introduced in parks starting in the mid-19th century, and now it occur in cities at densities 2.5 times higher than in rural areas. The coat color of eastern gray squirrels can either be gray or melanic, and this is caused by a 24 base pair deletion on the MC1R gene. The wild-type individuals are gray, and the melanic individuals can either be heterozygous for the 24 base pair deletion and a sort of more brown-black phenotype, while the homozygote individuals for the deletion are a jet-black phenotype. And previous work from my colleagues has shown that the prevalence of melanism decreases with increasing distance from the city center. But there could be multiple selective mechanisms at play explaining this decline in melanism that we're seeing. Melanism is associated with crypsis, and predation is the leading cause of mortality within the rural woodlands. And the wild-type gray individuals camouflage better within the secondary forest types in the woodland areas. So but predation pressure is reduced within the cities, and this could be not putting as much pressure on the melanism and increasing the amount of melanism we're seeing in the city. So this could sort of potentially be a relaxation selection. Alternatively, there could be advantages to melanism in novel city selection pressures. So in order to test whether novel selection pressure or relaxed selection, or both, are contributing to the genomic divergence between urban and rural populations, we used a focal study in which we sequenced 46 individuals at about 7x coverage for the whole genome. And within our urban population, we have a combination of melanic and gray individuals. However, within the rural population, the melanic phenotype is so rare that we didn't end up having any melanic samples within our urban system. So the PCA reveals some genetic structure between the urban and rural habitats. We have in the purple, the urban environment the urban samples are clustered rather tightly, and we don't see any separation between the morphs within the urban habitat. But there is a great more separation within the rural samples along both of the PC axes. And admixture plot does show a number of intermediate individuals, particularly in the rural samples, and this could possibly be due to gene flow, which supports previous work that's been done on that indicates that there is gene flow between urban and rural habitats. So in addition to our candidate gene for melanism, MC1R, we wanted to examine whether there were other locations across the genome that may be under selection or differentiated along. And so looking at an FST plot, we have our MC1R gene here on chromosome 16, and then we also, a number of these peaks were associated with a number of genes having to do with organ morphogenesis, olfaction, and sensory development. And we correlated this with significance to our PCA in order to sort of identify specific outlier regions clustered across the genome, though particularly clustered within chromosomes 5 and 6, in order to sort of to add to our sort of understanding of selection and our selection analysis. When we look at the urban and rural, we look at the melanic and gray morphs within the urban environment, we see that the the greatest FST peak is the window associated with MC1R. And in addition to that, we found that there was another peak that was occurring on that harbors a single gene, SMYD3, which is related to RNA transcription. So it's a little bit unclear how this may contribute to the genomic differentiation between the morphs. And so with these outliers in hand, we were looking at identifying selection, and we used ancestral recombination graphs and inferred topologies from there to distinguish between two selection scenarios, one in which there's early selection against gene flow, and one in which there's the more recent selective sweeps. And I'm going to walk you through an example of how we did this. So here's an example of one of our outlier regions on chromosome 6, and we use 3 summary stats to distinguish between these scenarios. First, we examine enrichment score, which is like the probability of individuals forming clades, and we would expect areas under divergent selection to have increased enrichment scores, but this could be due to recent selective sweeps or to islands of older islands that are resisting gene flow. To distinguish between those two, we looked at the time to most recent common ancestor, which we would expect recent selective sweeps to form more shallow clades, as you can see there in the purple, and then also cross coalescence times, which we would expect to be elevated in regions that have early selection against gene flow. So from this example here, this is an example of a region that supports recent selective sweeps for the urban environment in the purple. Overall, when we're looking at our outlier regions associated with our urban-rural comparison, about 73% of them showed increased enrichment scores. Less than 1% of those enriched regions had elevated cross-coalescence times, showing little support for sort of early selection against gene flow during population divergence, and more. Within this, the 29% of the enriched regions had low time to make. Were enriched for the urban environment and had low time to most recent common ancestor, indicating support for recent selective sweeps in these areas within the urban environment. And we did see some of the regions, about 14% of them, enriched in the rural environment and having a low time to most recent common ancestor, suggesting recent selective sweeps within the rural environment. When we take a look at MC1R, our gene of interest, we don't find enrichment in this area. So we don't find support for either of these selection scenarios. However, we don't necessarily expect for there to be positive selection. We're potentially wondering if there's relaxed selection occurring at this region. When we compare melanix and grays, we also see support for, or we don't see support for selection within this region. It was not enriched. Further down on the chromosome there, about 120 kilobase pairs away, there is potential for some potential support for some early selection against gene flow between the color morphs within the urban environment. So this prompted us to examine Tajima's D along MC1R. So we have the urban is purple and the rural is blue values for Tajima's D here, and the urban population seems to have a positive value of Tajima's D, which could indicate balancing selection within the urban habitat, and this would be consistent with the maintenance of 2 haplotypes within the urban environment, one with the deletion and one without. The negative value of Tajima's D for the rural environment doesn't tell us very much, because we only have 1 haplotype in that environment, the one without the deletion. So it's difficult to say what might have led to the reduction or the other haplotype to go away. So with that being said, if anyone has any ideas about how I might measure relaxed selection or identify what sort of purifying selection may be going on in this region, please come see me at the coffee break. And finally, just to sort of finish up, we have the only other peak that was associated with the comparison of morphs within the rural habitat, and that was the one associated with SMYD3 gene. And we see elevated enrichment along this gene and an elevated cross-coalescence time, supporting the idea of early selection against gene flow here. And so this also supports the idea that multiple modes of selection can be acting to explain the phenotypic divergence between morphs. So to sum up, our work adds sort of to the contributing, the growing evidence that there's the genetic basis for phenotypic divergence along urban and rural clines, and that multiple modes of selection can act and result in this genomic divergence along these clines. We found some support for local adaptation to the urban environment and some support for balancing selection occurring at MC1R that may be maintaining the two haplotypes that we're seeing in the city. And this work is part of a larger project that's and a number of collaborators, so thank you for being here today and listening to my small portion of it. Oh.
Yeah, can you explain what you mean by selection against gene flow?
So early in the sort of population divergence, the regions sort of, there was selection against gene flow at the sort of population radiation. Does that mean, sorry.
You're using two different kinds of terms of evolution, and so I'm not sure what you're trying to say there. Like, how do you select against gene flow?
So I guess there's limited gene flow in those areas, and then at the time of population divergence.
Okay.
Yeah.
Thank you.
Sorry.
A really basic question I might have missed in the introduction is this melanization of schools in cities something that's happened many times in lots of cities? Or
Yes. So the pattern of decreasing melanism has been noted in multiple cities. Yes.
And is there anything particular about these cities? Do you know the ecology of it, perhaps?
You should tune in on Sunday to a talk that's going to talk a little bit more about that. But we're working on it, and so yeah, tune in on Sunday for that one. Last one there, I think.
This was a really cool talk. Thank you for sharing it. I was wondering, I know that genes involved in melanin production can be pleiotropic with effects on immune functions, but I don't know what else they might affect in the physiology of squirrels. Do you have ideas or speculation around what other selective pressures might be playing a role in the balancing selection that you might be seeing in the city?
So I'm examining like immunity as well. So and sort of thinking about immunity. Some of the genes are associated with immunity. So but other things we're wondering about are sort of the thermogenesis and sort of understanding whether or not how melanism might contribute to thermogenesis. So and we're seeing sort of examples of things like muscular function and sort of angiogenesis that might contribute to that function.
Thanks everyone for coming. I'm currently a postdoc at the University of Chicago, but today I'm talking about work I did at my postdoc at UNC Chapel Hill with Daniel Matute and Dan Schreider on Aedes aegypti. So invasive species pose an increasing threat to human health. Models of future ranges under future climate projections predict that ranges of species like Aedes aegypti, shown on the right here, are going to spread towards the poles, so into new environments. And on top of that, global trade increases long-range dispersal, spreading agricultural pests and disease vectors across the globe. During these range expansions, populations go through a suite of demographic changes. So they go through population bottlenecks during the introduction. They experience expansions across the range after the introduction. And this also coincides with adaptation to novel environments. And these interact to influence genetic diversity in these species. This combination of demographic and selective pressures is hypothesized to contribute to an increased genetic load in the genome or a reduced efficacy of selection through the action of population bottlenecks and link selection. So this can result in a decreased efficacy of selection in these novel environments. So today I'm specifically talking about Aedes aegypti. Aedes aegypti is the most common vector of arboviruses in humans. And it was domesticated in Africa after the African humid period. So this resulted in increased desertification across the continent and a spread of mosquitoes towards human colonies in these drier environments. This led to adaptation to humans, including an increased preference to humans as a blood meal host. Following this domestication, Aedes aegypti is thought to have spread to the Americas during the slave trade. Previous work from Noah Rose et al. Has indicated here on the left that migration estimates between populations in the Americas and Africa seem to correspond to the number of slave ships about 300 years ago. And after the introduction to the Americas, aegypti is thought to have spread globally from the Americas. Within the Americas specifically, Aedes aegypti went through kind of a pan-national effort to eradicate the species. Then that led to several countries declaring that it was fully eradicated in the 1960s. Subsequent observations in these countries are thought to have been novel introductions, either from Africa or from the Caribbean, or from refugia within these countries. And because of this pretty substantial effort to eradicate aegypti with insecticides, it kind of unsurprisingly evolved insecticide resistance at multiple insecticide resistance loci and on multiple haplotypes, seemingly independently across the range. So given this complicated demographic history and this very strong selective pressure for eradication, we wanted to look specifically in the Americas and try to disentangle this demographic history and additionally look for any evidence for reduced efficacy of selection. There's really only two questions. I don't know why the first one's on there twice. So here we're using 131 samples kind of from across the range. So we have 3 populations in Africa, 3 populations in South America, and a number of populations in the US. This is a combination of previously published data, both from our lab and from other groups. So first, if we just look at how these populations are structured in PCA space, on the left, I'm coloring these populations by their country of origin. So in the warmer colors are populations or individuals from the Americas that seem to cluster kind of tightly together, with most of the diversity in PCA space displayed in Africa, kind of as you would expect with an introduced population. On the right, I'm only showing the Americas, and we actually see that our samples just cluster by population. So our South American populations on the right cluster together with themselves. And within the US, we kind of see these 2 broad groupings in yellow that I'm showing here. We can look more explicitly at population structure by modeling admixture in these populations. So here, if I model admixture including all of our samples, so African populations and American populations together, we find the best K, or the best number of admixed groups, is actually 2, and this separates the Americas in black and African populations in blue. This one African population that seems to be a mix is a Senegal population that seems to be a domesticated ancestry as opposed to more generalist ancestry in the other African populations. If we look at higher Ks, just for context of how this grouping is splitting out, there seems to be very little evidence for any admixture among these populations. Populations cluster into their own groups pretty consistently. But of course, a lot of this diversity in Africa is going to obscure some of the clustering within the Americas. So if we look just within the Americas, we actually again see the best K as 2 groups. But what's important here is that these groups don't seem to correspond to geography. So on the left is a population from Brazil that seems to cluster with some ancestry with a Colombian population. And this Colombian population seems to cluster separately from another Colombian population that's much closer in geographic space than the Brazilian population. If we look at higher Ks, the South American populations seem to be their own thing, whereas the US seems to split into these 2 groups, one that's seemingly independent from any other American population, and one group that seems to be admixed with some South American populations. I will note that these 2 big groupings in the US don't seem to correspond to geography as well. We have populations in California that seem to cluster completely separately, even though they're from the same city. But we have populations that cluster within California and Florida into the same cluster. Okay, so given this population structure, we wanted to look at the change in effective population size over time. And here we're just using these population groups as defined here in PCA space. So first, if we look at Africa, we see, so further back in time is towards me, further towards the present is towards the left. I'm showing effective population size over this time frame. We see shared bottlenecks that seem to correspond to the end of the African human period, about 5,000 years ago, and a slightly stronger bottleneck in green in Senegal that may correspond to this domestication history. But when we look at the Americas, we see really strong bottlenecks. So this is the exact same y-axis, where we're seeing reductions in NE from an ancestral value of about 600,000 down to like 10 to 30,000. So this is a reduction in NE of 90 to 95%. And this occurred seemingly separately in these populations. So the beginning of these bottlenecks don't line up with each other. Where they bottom out happen at different times. But they all seem to have occurred within the last 300 years, which lines up with this idea that they're introduced during the slave trade. The US here is being grouped as all of these populations, so this population structure is feeding into this lack of a recovery in the US. So if we actually split the US up into some finer population structure, we see that they seem to share this really strong bottleneck, stronger than in South America, that bottomed out around 100 years ago, with a very extreme recovery in the past few decades. And I'm just showing the full clustering there in yellow. Okay, so all those NE traces were done in SMC++, and with SMC++, we can also look at split times between these populations, assuming a clean split model, so no migration. So every split time that I'm showing here is biased towards the present if there has been migration between these pairwise populations. So in the yellow points that I'm showing here is the most important part. These are the split times within the Americas. And all of these split times correspond to more than 1,000 years ago. So these split times far predate the introduction during the slave trade, telling us that populations within the Americas were already deeply diverged within Africa before they were introduced to the Americas. So this seems to be an introduction of multiple ancestral lineages into the Americas rather than a single lineage that then spread throughout the Americas. Okay, so given this really extreme demographic history, we can then look at genome-wide diversity. So if we look at just genome-wide neutral diversity, I'm using the same colors here, so warm colors are the Americas, cool colors are Africa. We see a reduction in genome-wide diversity from about 3% to about 2%. So this is a reduction of about 35%. And this is perhaps not that big of a reduction in diversity considering a reduction in NE over this time of about 95%. So the American populations seem to be surprisingly diverse given their extreme demographic history. And we can then look at measures of the efficacy of selection instead of just neutral diversity. So here I'm showing pi naught over pi 4, which is the same thing as non-synonymous versus synonymous diversity. So higher values indicate a relaxed efficacy of selection. As you can see, these overlap quite substantially, but we see a very subtle increase in pi naught pi 4, indicating a slight reduction in the efficacy of selection in the Americas. This estimate is sensitive to non-equilibrium dynamics, so we can look at this a different way using the distribution of fitness effects of new mutations. So this is looking at the proportion of the genome corresponding to nearly neutral mutations on the left to increasingly deleterious mutations on the right. So if you just focus on that leftmost bin of 0 to 1, this is our nearly neutral mutations. Again, in the warm colors, we see a significant but very slight increase in the proportion of nearly neutral mutations, corresponding to a slight decrease in the efficacy of selection. And just one final thing. On the left, I'm showing the genome-wide efficacy of selection as shown by pi naught pi 4. But if we look around known sweeps, and these are sweeps detected with Sweep Finder from a previous postdoc in the lab, which I'm showing these dashed lines on the right and the triangles, we see a much stronger signal of this reduced efficacy of selection around sweeps, which is a sign of linked selection in the genome. There is a greater proportion of the genome that's experienced recent sweeps in the Americas relative to Africa, so some of this genome-wide effect of decreased efficacy of selection might be due to this draft effect in the genome. Okay, so just to wrap it up, population structure in Aedes aegypti in the Americas seems to be strong at both coarse and fine scales. We see population divergence between samples within the same city that's thousands of generations, thousands of years. We find little evidence for admixture among these populations, and these American populations seems to be divergent ancestral lineages, and we see a very small reduction in the efficacy of selection, and that's all my time. Thank you.
Okay, hi, I'm Melanie. I'm a postdoc in Helsinki, and I'm going to talk about genetics and colour in the tiger moths. So I'm just going to start by introducing our study species. So this is Arctia plantagenus, the wood tiger moth, and it has these bright colours on its hind wings, which it uses as a defence to deter predators. And in the males, they have this polymorphic colour, so we find both these yellow and white males. And the colour is also sexually dimorphic, so the females are red, and they also have variation in their colour, which is more continuous, so that goes from kind of yellow, orange to a darker red. And this is how we find them in the wild. So colour in general is a great trait to study because we can look at it from many different contexts, from sexual selection, signalling, thermoregulation, and camouflage. And such as in the tiger moth, we can find multiple colour phenotypes within the same populations, and these are known as colour polymorphisms. And how polymorphisms are maintained can be quite puzzling because colour is often used as an aposematic trait, so as a warning signal to predators that a species is toxic. So we would expect predator learning to lead to convergence on a single colour or pattern, but in fact these types of colour polymorphisms are quite common in nature. And they can be maintained by balancing selection, where we see genetic correlations between the colour locus and other traits which have benefits in different contexts. So in our focal populations in Finland, we find both of these white and yellow moths within the same populations. And from a long history of breeding these in the lab, we know that hind wing colour is a Mendelian trait controlled by a single locus with two alleles. And the white allele is dominant over the yellow, so the heterozygotes are also white. But what is interesting about this colour trait is that these genotypes also have differences in behavioural and life history traits. So the morphs have differences in their predator avoidance strategies, immune systems, blight ability, and chemical defences. So, for example, the yellow moths have increased defences because of their chemical defences, whereas the white morphs have increased flight abilities and immune systems, so they each have different advantages in different situations. So my question has been, what is the genetic basis of this polymorphism? So complex polymorphisms where we have multiple traits involved are often explained by supergenes or chromosomal inversions, where we have multiple alleles kept tightly linked together in the genome without recombination breaking them apart. So that's the case in this red pole, where genes for beak shape, carotenoid, and melanin production are closely physically linked in the genome. And we also see inversions in deer mice and in Heliconius pneumata butterflies, where supergenes produce this variation in wing colour patterns. But there are also some cases where these complex polymorphisms have a more simple genetic basis. So this is the case in the wall lizard, where colour genes have pleiotropic effects on behavioural traits. And so to look at this in our moth, we use 3 different data sets. So we started with some crosses between heterozygous females and homozygous yellow males. And then we used the white and the yellow male offspring from these crosses to do a QTL analysis. So looking for correlations between the hindwing colour and markers in the genome. And this showed this really clear single peak, which was on scaffold 206 in our reference genome. Then we used individuals from wild samples to do a genome-wide association study. And again, this showed just one clear peak, and also these top SNPs were on this 206 scaffold. And there are just 2 SNPs above the high significance threshold, and these were just 150 base pairs apart. So because these are so close in the genome, we're not really seeing any evidence for a supergene or an inversion, which would be seen as a much wider region of the chromosome. So it seemed more likely that we have a simple genetic base for this trait. And if we look at what is in this region of the genome, so the location of that QTL peak and those top SNPs, we find that there's a group of yellow family genes. And the yellow family of genes in insects form part of the melanin synthesis pathway. And we've had other work in the lab which has shown that the pigments on the moth wings are melanins. So the coloured parts of the wings are pheomelanins and the black parts are eumelanins. So it makes sense that we found these melanin genes. And in particular, this gene yellow E has in the past been associated with larval colour in bombyx moths. So this looked really promising, but we didn't find a perfect association between this yellow E gene and the hindwing colour. So next we had long-read data for both a yellow moth and a white moth. And we aligned these to look for structural variation rather than individual SNPs. So we looked for any insertions, deletions, or smaller inversions. And what this showed us is that in the white morphs, they have a duplication in this region, which is about 100 kb long, and it includes a second copy of this yellow E gene. So the white morphs have 2 copies of this gene, whereas the yellows only have 1 copy. And then what we wanted to do was to get some functional evidence that this is the gene we were looking for. So we used the CRISPR-Cas9 system to design a guide to knock out this duplicated copy from the white morphs, and in theory this would turn them yellow. So this is what happens. So at the top we have the wild-type white and yellow morphs, and at the bottom the CRISPR knockouts, which are from the white stock that we have in the lab, but you can see they have yellow wings now. And then this is just a video to show that behaviorally they didn't seem to be acting any differently. There was no other morphological effects, so it was only the colour that seemed to have changed. But one other thing that we did find was that the UV reflectance also differed. So in the wild-type white morphs, they reflect quite brightly UV from the hindwings. So in the pictures on the right, the bright pink areas are where the UV is reflected. But if we see in the CRISPR knockout at the bottom, we've kind of lost this UV reflectance. So one thing we don't know yet is whether this is due to a change in scale structure, which could have knocked out the UV reflectance, or whether it's more that the yellow pigment is masking the reflectance of UV. So I next wanted to introduce the second part of this project, where we're looking more generally at population genomics and how this allele could have evolved and spread through different populations. So the moth is very widespread across the northern hemisphere, and we find different frequencies of the different morphs across populations. So as well as our populations in Finland, where we find both the male white and yellow morphs, we can also find monomorphic populations. So for example, this population in Scotland, we only find yellow males. In Estonia, we find mostly white males, but with yellow males at about 3% frequency. And then in the south, we have this interesting population in Georgia, where the sexual dimorphism isn't found, so both the males and the females are the red morph. Usually we only see the females as the red morph. And so we have this long-term data set where we have been collecting moths from 4 populations in Finland every year since 2008, and looking at the frequency of the white versus yellow males that we find. And in 3 of the populations in northern Finland, central Finland, and southeastern Finland, we find the white morphs are more abundant, with the yellow morphs being between 25 and 40% frequency. But we have this quite interesting population in southwestern Finland, which is just around 150 kilometers away from the southeastern population. But here the yellows are much more frequent, so we find the yellow morphs at around 75%. So we have sequenced a subset of these time points across all populations, and I've just started to look at population structure. So the PCA and admixture plots show that the samples group quite nicely by geography, but the interesting thing we find is that there's quite a big difference between the southeastern and southwestern population, despite the fact that there's no geographical barriers between them. So one hypothesis that we haven't tested yet is that the southwestern population, which is the yellow population, is more closely related to the monomorphic yellow sites that we find in the west, whereas the southeastern population, which is the more white population, could have come from Estonia, which is where we see mostly white males. And then we have some measures of genetic diversity, which show that in particular in the northern Finnish population, there seems to be a more recent population expansion, suggesting that this moth is moving northwards. And phenotypically, the moths we see in the north have more melanized wings, so they have more black on their wings, which makes sense when we're thinking about melanin in terms of thermoregulation. So we see this pattern in quite a lot of species. And so there's a lot that we want to test with this data. So looking at population expansions and contractions, tracking allele frequencies across the different time points that we have. And then I also want to see if there's any signatures of balancing selection around the color locus, which could help to explain why we do see this polymorphism. And then also comparing males and females, particularly thinking about migration, because we know that the males are much more active than the females and they travel much further. So just to summarize what we have, the color genotypes in males also have differences in their behavioral and life history traits, and we found that this duplicated gene is associated with white males. But one thing we don't know is how this gene duplication could also be controlling other traits rather than color. So it could be that there's a regulatory element which is controlling color, but also controlling other traits via other genes. And our future work will look a bit more at how this gene has evolved and the demographic history. So I'll just thank the MAPES lab in Helsinki and Jyväskylä, and this was also a collaboration with a group in Cambridge as well. And thank you for listening. So it's quite similar. So the original copy, I think it has 7 exons, and then the duplicated copy has the first 5, so it's kind of lost some of the exons, yeah.
Great talk, thank you so much. You said that the individuals who had that second yellow E knocked out had an altered UV reflectance. Do you see that reduced UV reflectance
So in the wild, the yellows don't have the UV either, so it's only the whites. Yeah. Yeah.
In your classes, is it possible to do a sort of formal linkage analysis of the pigmentation of behavioral traits? Like, are some of them very rarely doing the unexpected behavior or anything like that?
So we have some more data that's hopefully going to look at this. So we've collected data from 6 families and tried to get as many traits as possible. And then we're going to do a QTL analysis to see if there's any links between color and many of the other traits. Yeah.
Thanks for being here. My name is Maria Akopyan. I am a postdoc at the University of Southern California. I'm excited to tell you about some of the work I've been doing for the last several months, looking at demographic histories and recombination landscapes of divergent fox lineages. I'm also going to talk a bit about reference bias. I didn't realize that when I made the title of the talk, so there's a little change there. But seems kind of silly to open this way, but we all know this, that access to genetic data has really revolutionized the field of evolutionary biology. Thanks to advances in sequencing technology and the plummeting costs, we can now investigate the genetic patterns associated with fundamental evolutionary processes. Genomic data is often used to identify populations, assess genetic diversity, reconstruct evolutionary history, identify the targets of natural selection. And our ability to use genomic data for evolutionary inference in most non-model organisms is dependent on important genomic resources, such as reference genomes. So reference genomes serve as coordinate systems for referring to and annotating sequence features, making them essential tools in genetic studies. So you can generate cost-efficient short-read sequence data for your natural populations of non-model organisms, and then map that to a reference genome, which allows for comparisons of individuals and populations. And in the absence of a species-specific reference genome, researchers often use genomes from closely related species, but this can introduce reference bias. And reference bias is when reads containing non-reference alleles fail to align to their true point of origin. And so this causes genomic variants to be missed or miscalled, and it affects mapping and variant calling and downstream analyses. And while there is evidence that reference bias can lead to overestimated heterozygosity and inaccurate detection of structural variation, few studies have actually examined how it may affect the characterization of historical demographies and population recombination rates, which are efforts, I would argue, are growing in the field of evolutionary genomics. Oh man, my fonts got messed up. So the gray fox actually provides a really compelling case to examine the impact of reference bias on the inference of population genetic variation. So just a bit about the gray fox. They're widely distributed, ranging from southern Canada to northern South America. They're pretty common but elusive. They have climbing adaptations, and they're really cute. And there's some evidence that there's divergence between the east and west lineages in North America. And gray foxes are actually sister to all other living canids. So note that fox is just a descriptive term. There isn't a single lineage, but at the top in the red is maybe the traditional fox lineage you would think of, but that's where the red fox is and the Arctic fox is. But then there's also like a green lineage of the South American foxes. And then down here at the bottom in gold, where I'm showing you an arrow, you can see the lineage that includes the gray fox and then its dwarf descendant, the California Channel Islands fox. And then right above that in the blue lineage is where the dog and wolf clade is. And dogs are obviously a much more recent clade, and they've been domesticated like crazy. And the reason that I'm highlighting dogs is because previous gray fox genetic studies have relied exclusively on a dog reference genome. And this reliance on the dog reference is quite precarious. So the dog genome lacks synteny with the gray fox. Dogs have 39 chromosomes compared to the gray fox's 33. And so these large-scale karyotypic changes likely introduce bias in analyses, especially those that rely on synteny. And so here, leveraging this recently published gray fox reference genome generated from an individual from Vermont, which is shown by the gold star on this map, we reanalyzed some whole genome sequencing data from 2 populations of North American gray foxes, previously published by Kirepka and colleagues. And we included 26 individuals 14 from the east, which are shown in gray, and 12 from the west, shown in the black. So here we wanted to examine the impact of reference bias, focusing on these 3 reference genomes. CanFAM3, which I'll be showing in red, it was the reference genome that was used in the previous gray fox studies. And then we also looked at CanFAM4, which is a more contiguous updated version of the dog genome to see if an improved assembly makes a difference. And then, of course, looking at the gray fox reference. And so using these 3 reference genomes, I'm going to examine how our inference of population genetic variation, demographic histories, and recombination landscapes are impacted. And building on that and assuming that the gray fox reference genome is providing a more accurate framework for aligning and interpreting genetic data, I'm going to explore these same parameters, but here just focus on comparing the eastern and western lineages, given our ability to more reliably estimate these population genetic parameters with a species-specific reference. So let's start with looking at metrics of diversity and differentiation, pi and FST. So remember that pi is average pairwise differences between all possible pairs of samples of individuals in your sample, and it measures genetic variation. So higher levels mean there's higher genetic variation. FST is a measure of differentiation, and higher levels there means there's greater differentiation between populations. And we see that both pi and FST are quite different when estimated using the different reference genomes. So with dogs, we see that, with the dog genome, we see that nucleotide diversity is estimated to be 5 times higher in the east and 7 times higher in the west. And FST is less than half with the dog reference genomes. And then furthermore, you might expect a negative relationship between pi and FST, as typically the evolutionary phenomena that might reduce variation also will increase differentiation between populations. And so when we look at correlations between pi and FST, we see that there's a positive relationship only for the western foxes with the dog genomes. And typically, this type of pattern can be interpreted as evidence of secondary contact, which interestingly has been proposed to be happening with, in Western foxes. But again, this was all based on the dog genome, so that's something I'm going to look into a little bit more and see what's happening in the West. Then looking at the site frequency spectra for each population based on the different reference genomes, the red and the blue, again, are the dog reference genomes. And you can see that you have higher singletons or more rare variants compared to the gray fox, and this pattern is more pronounced with in the east. And there are more common variants in the east and when mapping to the gray fox genome. And remember that the gray fox genome is originating from an individual from the east. And this is quite alarming because many of our population genomic inferences rely on the SFS, like demographic inference. And sure enough, estimates of effective population size measured as the inverse of the coalescence rate are vastly different when estimated using the gray fox reference, about an order of magnitude higher than estimates based on the dog genome. And then the changes in effective population size also appear to be more rapid and recent with the dog. And there aren't really, like, you don't do that much better with an improved assembly with the dog genomes. And the extent of this pattern is both surprising and concerning. I'd be curious to repeat this with other studies to see if this is something that's just funky happening with gray foxes. But yeah, I find it quite troublesome, especially when you're using this information as a prior for estimating other population genetic parameters like recombination rates. And here we estimated population recombination rates, which is, takes in that demographic information and based on measures of LD can give us an estimate of recombination. And here we see the overall, we resolved much higher rates of recombination when using the dog genomes and compared to the gray fox. And also, when we look at the relationship with chromosome size and average recombination rate, typically smaller chromosomes tend to have lower recombination rates, and we see this pattern in the gray fox, but that doesn't hold with the dog genomes. And so given this pretty intense impact of reference bias on these estimates, I'm going to focus on describing some of these patterns in the gray fox, just focusing on what we see with the gray fox reference. So looking at genetic differentiation across the genome, it's elevated genome-wide with an average of about 0.4 between east and west. I worked on fishes before this. I've never seen an FST that large. What? So yeah, highly differentiated lineages. And if we look at genetic diversity, we see that average nucleotide diversity in the west is about twice than what we see in the east. And then on the bottom, I'm showing you the smoothed values of pi across the genome. And the landscapes are pretty similar between the populations, but overall, you see west is higher. And there's this pattern where it seems that diversity increases near one end of chromosomes. Then looking at the demographic histories, we see that there are larger population sizes in the west, and there's a prolonged collapse in the east. And I wanted to compare the SFS of the two populations, so I dropped 2 individuals from the east, so I had the same number of 12 individuals. And here we can see that we have more rare variants in the west and more common variants in the east. And then looking at the recombination landscapes, we see overall recombination is higher in the west, and similar to how diversity was peaking at one end of the chromosomes, we see that recombination landscapes have a similar pattern. And another thing that was resolved with the gray fox reference genome was confirming that PRDM9 was lost in this lineage. So if you're not familiar, but PRDM9 is responsible for recombination hotspots in some mammals, including humans and mice. And it's known to be a pseudogene in dogs, and the recombination hotspots then tend to be concentrated in these CPG islands and transcription start sites. So the bottom, I'm showing you this pedigree-based recombination map of dogs, and then on the top is the population-based recombination map we resolved from the gray fox, which we also know doesn't have PRDM9 and has a pretty similar landscape of recombination, which is pretty reassuring to see. So I've gone over quite a bit there. Just to summarize, reference bias can really skew your allele frequency distributions and lead to underestimated population sizes and overestimated recombination rates. And then we have these really divergent lineages of gray foxes with varying demographic histories. There appears to be more foxes and diversity in the west, and the recombination landscapes are similar to what we see in other canids. All right, so I have to thank my awesome co-authors and you for listening, and I can take any questions. Yeah.
Hi, really fascinating stuff. So I'm curious if it would be possible to dig into the reference bias a little more and see if there's like highly synthetic regions. Do you have more comparable population genetic stats versus less synthetic regions?
Yeah, we do a little bit better with, I've done it like at a chromosome scale, just like chromosomes that are like over 90% synthetic or something, and the pattern is more similar with, yeah, for sure. But I want to dig into that a little bit better. Yeah, that's a good question. Yeah?
Is it possible that you could get a similar reference bias on a smaller scale if the reference genome is more similar to the east population than the west population?
Absolutely, yes. There was actually a stickleback reference bias, because I mean, I guess we don't really have like many non-model organisms where we have multiple reference genomes from multiple populations, maybe not so much now, but definitely before. And so, yes, you can definitely have reference bias within a species too. So it'd be great if we could get a Western gray fox genome.